Yeah, thanks for giving me the opportunity to speak to you all today. I'm going to talk about a resource called ClinPGx. It's a Clinical Pharmacogenomics Resource. It's somewhat rebranded PharmGKB, and it's a combination of PharmGKB and CPIC. And I'm going to talk about our work in pharmacogenomics, our work with drug labels, and some of our interactions with the FDA. So just to give you a little bit of background what I'm talking about here. So PharmGKB as a pharmacogenomics knowledge base was originally established in the year 2000 as part of the NIH-funded PGRN. It was designed to be a database to hold the phenotype and genotype information created from other sites within the PGRN originally, but also a knowledge base connecting information together from these resources and also information that we curated from the peer-reviewed literature. So about 9 years after PharmGKB was started, CPIC began, the Clinical Pharmacogenetics Implementation Consortium. It was a joint project between PharmGKB and PGRN. And the goal of this group was to create guidelines for clinicians to be able to understand how to use pharmacogenomic data, information on their patients that they might have in hand. How can they use that to prescribe medication? And part of the reason why this group was started was because at the time, especially, the information on the FDA-approved drug labels with respect to pharmacogenomics was scant, and the information that was there was not very directive per se about how exactly a clinician should use that information. So these projects went on for many years, a decade or more. In 2021, we proposed ClinPGx as a way to bring all the different PGx resources under NHGRI umbrella together. And we established, the website was established just last summer. So in this talk, you're going to hear me talk about PharmGKB, CPIC, and ClinPGx. Just know that it started for many years. There was PharmGKB, and now we call it ClinPGx, and it incorporates CPIC as well. We're really excited about ClinPGx because not only does it bring pharmacogenomics resources and some tools together, but also looking forward to working with ClinGen to align the pharmacogenomics area with genomic medicine in general. And so we'll be working with ClinGen, and we'll actually have applied as an affiliate grant, ClinPGx, with the ClinGen group. So this slide is just to show you that we've been interacting off and on with FDA for many years now, going back as far as 2010. We had some initial conversations with FDA about the pharmacogenomics on their drug labels. What information was there, what information was missing, that sort of thing. They had a table, they still do, a table of pharmacogenomic biomarkers that's available on a website. And it has many drugs on it. I'll tell you a little bit more about that in a few minutes. And we began curating the labels to extract the pharmacogenomic information from these labels around 2011, 2012. We've had interactions with the FDA over time. One of the things more recently that we've worked with PharmGKB and CPIC submitted applications to become a recognized human genetic variant database, and I'll tell you briefly about that in a few minutes. So just start with this table of pharmacogenomic biomarkers. In the year 2010, there were about 200 entries on it. It's exploded to over 650 entries today. But even in 2010, those 200 entries, many of the drug labels on there didn't really have actionable information for how a clinician could use pharmacogenomics on the label. But there was a big misconception about that, and a lot of people misconstrued this table and would say, oh, there's, way back over a decade ago that, oh, there's already 200 drugs that we can use for pharmacogenomics, and that just wasn't true. So we really wanted to go in and help people identify what labels on this list were really actionable. So we, like I said, began curating these around 2011, 2012. So The goal of our curation of these labels was really to help them understand which ones contain gene or variant information that can be used for prescribing these drugs. And so we asked a few questions to be able to separate these drugs out from the others on the list, and came up with kind of like a criteria and like a tagging system for them. So the first one was, does a clinician need to have a genetic test result prior to prescribing this drug? This was a big question at the time in 2010. And so some labels do say that there needs to be some kind of genetic test or some kind of biomarker tested in order to prescribe the drug. There's not tons of these that are what we consider classical pharmacogenomics labels, but they exist. There's also some labels that kind of hedge the wording a little bit and just say like people should consider a genetic test before prescribing. The next question we wanted to know is there genetic information on the label that can affect prescribing outside of testing? So actually there's a good chunk of labels where they don't discuss testing at all, but they do say that if you know that your patient is, for example, a CYP2D6 poor metabolizer, you should decrease the dose or prescribe a different drug. And so that's what we mean by actionable pharmacogenomic information in this case. Doesn't discuss testing at all, doesn't touch that, but does say if you know something about the genetics of your patient, you can do something different with prescribing the drug. The next question was, is there genetic information that the clinician could or should keep in mind? So maybe they don't give actual direction on the label about what to do, but the label might say something like CYP2D6 metabolizers have higher plasma concentrations of this drug, and leave it at that because the drug is metabolized by CYP2D6. So in this case, there's no actual direction for a clinician what to do, but a clinician could put two and two together. And if they already know that their patient is a poor metabolizer, and they know that, that CYP2D6 extensively metabolizes this drug, and there's an alternate available, they might decide, I'm going to start with a different medication first instead of trying this medication. So in that way, it's informative to a clinician, but not necessarily directive or actionable. And then finally, there are some labels on the table where the genetic information just does not really help with implementation at all. For example, the drug label might mention that in one of the clinical trials, there was a section of the population that had a particular genetic biomarker, and then leave it at that. They don't say if there's something different with that group, if they had more adverse events or better efficacy of the drug, but they just kind of leave it at that. And so there's a fair number of labels on the FDA biomarker table where they mention a gene, but not really anything more that you can do with it. And then there's another group where they might actually say CYP2D6 is not clinically relevant for this gene. So it's also on that table, just like all the other ones, and you can't really tell just by looking at that table that in this case, maybe they're saying that there is no pharmacogenomic information that would be actionable. And then finally, we wanted to, at PharmGKB, be able to tell people if there is relevant genetic information, what specifically is it? This is the actionable prescribing information. And so we extract that from the label and present that up front to users. So I'll show you an example of a curation in a minute, but I just wanted to first show you kind of a graph that talks about the labels that we've curated to date under all those different kind of categories that I just went through. And you can see that there actually are actionable ones where they talk about really doing something different based on a patient's genotype or phenotype. Is actually kind of a small number compared to all the labels on the table. And this testing required, it looks like there's a ton, right? But there's mainly this gray bar here. And that's because what we're indicating here is that over 100 of these kind of labels that talk about testing are those with companion diagnostics, looking at kind of tumor-related genetics, oncogenes, that type of thing. And so those aren't what we consider traditional pharmacogenomics, in that you're changing the drug prescribing based on germline variants in the patient. There are also drugs in that group where the drug is prescribed prescribed for a genetic indication. So, for example, a drug that's used to treat familial hypercholesterolemia might be listed on the FDA's table because they'll say LDLR is a biomarker. So again, not really what we consider traditional pharmacogenomics. So it's I think, a useful resource for people interested in knowing what kind of actionable information exists today on the biomarker table, labels on that table, by looking at our resource at ClinPGx. This is just an example of what it looks like on our website for example, the label for abacavir. We just have some tags to help orient you right away that yes, there's some kind of information that testing is required and an alternate drug should be prescribed. And it's regarding, we have in this kind of yellow box, what that information is. If a patient has HLA-B 5701, for example, they should be tested for this variant before prescribing. And if they have the allele, not to prescribe this drug. Just a different example for atomoxetine. This is an example of a drug where it doesn't mention testing really at all on the label, but it does say that there's something that you can do. And I highlight this label here because we also at ClinPGx are very focused on any kind of pharmacogenomic information we can find that's relevant to the pediatric population. So it might be of interest to this group. And so we tag all of our curations, whether it be from the literature or drug labels, if we can find genetic evidence for pediatric populations on it. And this is one example where, we're talking about in children. What they're saying basically is that 2D6 poor metabolizers should start their dosing at a different dosage than others. This is just an example of one where, 2D6 is mentioned, talks about plasma concentrations, but doesn't give any kind of direction. You can see that big yellow box there is missing because there is no actual prescribing information on the label. So that's kind of the range of drug labels that we've worked with the FDA to, we curate information from that table, but we have also identified labels that haven't been on the table, especially back when we started, back around 2011 to about 2015. We had conversations with the FDA back and forth to try to update them when we found labels with pharmacogenomic information. They would update their table and likewise notify us when the table was updated. Those conversations have kind of petered out over the years just because they have a more systematic way of updating their table now, but we do still have some communications with the FDA, but we also use AI to monitor now changes to the table and changes to drug labels as well. So in 2020, FDA came out with a separate table. This table is a table of pharmacogenetic associations. I don't know if you guys have heard of this, but it sometimes gets conflated with that biomarker pharmacogenomics table. This table was really created by a different group at FDA, and basically they look at drug-gene interactions, and they've separated their table into 3 sections. Drug-gene interactions for which they think that there's something actionable, the recommendations can be made. A section for drug-gene interactions that they think have potential impact on safety or response, and then a table where they believe that the evidence is focused on PK only right now. And we curate this information as well. As I said, this comes out of a slightly different group at the FDA than the other table, and so the information largely agrees between these tables, but not completely. There are some discrepancies between them, and so we curate this information, allow people to compare, and I'll show you that in a second. Importantly, FDA doesn't connect the actual genotypic information or the variants to the phenotypes, and it's the phenotypes, the wording that's mainly on those tables and labels, right? They talk about poor metabolizers, but don't tell you which genetic variants make you a poor metabolizer. So it does make it a little bit difficult for people to try to implement off of the labels directly. And ClinPGx, primarily through the CPIC guideline process, does provide that mapping from which alleles have, affect a patient's drug response, and how you can map the individual variants to a phenotype like a rapid, normal, or poor metabolizer. And so we can use that information, that's gene information that's created from those guidelines, and apply it to this drug label information, even if there's no CPIC guideline for that particular drug. We can use that information to provide tools or even EHR alerts to users. And this is just a screenshot of a tool that's available on ClinPGx, where you can enter genotypes for any particular gene. In this case, simulating entering a CYP2C19 star 1 star 3. You enter that information and the tool will tell you CYP2C19 star 1 star 3, if that's your genetic test result, you are an intermediate metabolizer, or your patient is. And then I don't expect you to read everything on the screen, but this is kind of the results that we pull back. We use that intermediate metabolizer label, and we go in and we look at all the labels from the FDA that we've curated. That's what this column is, that might be have some kind of information for CYP2C19 and an intermediate metabolizer. This column is the tables that I showed you. Like I said, slightly different information. There's information from CPIC and information from, oh, I thought I was, sorry, I thought you could see the mouse on my screen and you cannot. So the, let's see if we have the pointer, oops. Okay, I don't know the pointer. Okay, so the last column would be the table. The second to last column is the information we've curated from the labels. Then the first two columns are guidelines. One's CPIC guidelines and one's from another group. And you can see just like really, the yellow tags alert you to if there's something that you can do, some actionable prescribing change that should go on. The gray tags means not really. And you can see right here that there's a difference between what we found on the labels and what we found on the table, and that's just wording. In the tables, they talk about, they often group intermediate and poor metabolizers together, versus the wording on the labels is generally just looking at poor metabolizers. So there are some differences there. See if I can advance. Similarly, you can use the same kind of information in the EHR system. This is just an alert. It's actually regarding a G6PD and a CPIC guideline, but we can and have done the same at Stanford for information from FDA drug labels that don't have CPIC guidelines as well. And if you're familiar with the EPIC system, you can use this kind of information in your genomic indicators. I'm not going to go into that. So just a couple of words, switching gears, just a couple of words about this recognition of genetic variant databases. I mentioned that CPIC and PharmGKB submitted to this. So in 2018, FDA issued some guidance about using databases to support in vitro diagnostics. And so they put out this this call for databases to apply to be recognized. It's an arduous process. A couple have been recognized so far, ClinGen and OncoKB. These two databases are largely regarding genetic variants' effect on heritable disease or cancer. CPIC and PharmGKB obviously are talking more about drug response, which made it a little bit more complicated interactions with the FDA. We had multiple meetings and have discussed in detail the CPIC guidelines. Are very familiar with them and our work with the FDA labels. And we're really looking forward to our collaboration and our being part of the ClinGen family and working with the FDA further under that umbrella, especially seeing as how ClinGen has already gotten recognition of their database. Okay, so I'm just going to spend a few minutes now on the question I think the organizers really wanted to hear about how can we really affect change with FDA-approved drug labels. Is there have we been able to in our discussions about comparing and contrasting what's on the label, what kind of evidence we find in the peer-reviewed published literature, and what kind of guidance can we give in the CPIC guidelines, can we change what's on drug labels? And not to be the bearer of bad news, but in our experience, it really takes a lot of dedicated advocates, time, patience, and persistence to affect any kind of change on labels. And I have a singular example to give you, and this is good news because I'm giving this talk today instead of 6 months ago, because it would be different. So fluoropyrimidines and DPYD is an example. CPIC first published a guideline in 2013 that recommends changing the dose, lowering the dose, or prescribing a different drug for patients that have deleterious DPYD variants, depending on what variants are present in the patient. When prescribing capecitabine or fluorouracil. At the time in 2013, there was no information, no wording about testing on the DPYD about DPYD on the drug labels. And the NCCN guidelines at that time actually said they did not recommend testing for DPYD. Over the years, we had this, so like I said, in 2013, we started advocating for this. Over the years, by 2020, there was a patient advocacy community that had started to grow. Unfortunately, from relatives of patients who had passed away due to adverse reactions to these medications, and these people carried these, had these DPYD variants. So there was a huge kind of patient advocacy community that grew, and a really public lawsuit in 2022 brought from a widow of a patient that unfortunately passed away. So this really drove more public recognition of the issue than I would say that we were able to through CPIC and PharmGKB, but building on information that we started publishing as far back as 2013. So after that time, we have continued to revise the guidelines. We revised them at least once, and now they're under revision again, and will be coming out hopefully in the next year. And together with other PGx experts in the field, we really started calling for talks. I say we as a PGx community, not we as ClinPGx. The PGx community really started pushing for talks with FDA and NCCN by 2023, I would say. This was a big topic at the PGRN annual meeting in 2024, and there were FDA representatives present there. Not that this was news to them in 2024, because just to be clear, there are FDA representatives, part of CPIC, sit on CPIC calls and what have you, so they're very familiar with the guidelines. By 2025, FDA and AACR hosted a DPYD testing workshop. They brought people together to look at like what's the landscape and what's the current approaches to testing and so forth. And then NCCN did update their colorectal guidelines in March of 2025, and you may not be able to read it, but I've kind of highlighted it here. There was a footnote in the guidelines that said, routine DPYD testing prior to fluoropyrimidine therapy is not recommended. And there's now a line through that. It's like scratched out. And it says next to that, testing for DPYD genetic variants should be considered prior to fluoropyrimidine therapy. And it goes on to talk about physicians should be discussing that with patients. So that was huge. And then the pièce de resistance is that in October of 2025, the FDA did update the wording on the capecitabine label to say that there should be tests for patients for genetic variants of DPYD prior to prescribing this drug when possible, and to avoid using the use in patients with deleterious DPYD variants. Again, not specifying which variants those are, but that's fine. We've already taken care of that with CPIC and ClinPGx, and work to expand that list all the time. And then in February 2026, the fluorouracil label was updated as well. I just want to say that I'm trying to change actual drug labels is not an easy process. I'm not aware of any kind of there's no really great way to do this. We still know of multiple cases where the evidence right now for pharmacogenomics does not necessarily match up with what's on the drug label. This example is pretty unique, but that's why we have the CPIC Consortium and continue to publish those guidelines to help try to get that information out there. And I just want to say that this, of course, this is not due to any kind of lack of the scientists at FDA are aware largely of what's happening, what's going on. From conversations with them, it's everyone's resource limited, and not just us, right? FDA as well, and changing and updating drug labels is a big process. It's not easily done. So it's a rate limiting step, and I hope that there are some processes in the future to do this, but currently they're not really great ones in place. And I just want to say thank you to the entire ClinPGx team, specifically the other MPIs on this project, Terry Klein and Kelly Caudle, and our entire teams at Stanford and St. Jude's. And if you have any questions that you think of later and you'd like to ask, please feel free to send an email to feedback@clinpgx.org. Thanks.